So welcome to our workshop on multimodal analysis of CITE-seq data with CiteFuse. My name is Hani Kim, and this workshop will be co-led with Yingxin Lin, who is also here in the Zoom room. So if you already haven't done so, please follow the login steps shown on the screen to access the workshop material. So I'll give a few seconds for people to do that. And perhaps Yingxin could help me copy and paste this info onto chat as well. Okay, so and during the workshop, whenever you have questions, please feel free to unmute yourself and ask questions anytime during the workshop. So we're very relaxed. Or alternatively, you can also leave a message on chat to everyone or privately. Okay. Just another brief introduction about us. So we are both third-year PhD students at the Department of Mathematics and Stats at the University of Sydney in Australia. And we're both part of the Sydney Precision Bioinformatics group. So if you're interested in more of the work that our group does, please follow these links to see more details. Okay, so the setup and schedule for today's workshop is a brief introduction on CiteFuse and CITE-seq data. And then we'll move on to performing the analysis using CiteFuse, of which there are two major parts: the pre-processing and integration of CITE-seq data, and then all the downstream analytical components of CITE-seq. And then we'll wrap up with conclusions and Q&A. And again, if at any point people have questions, please leave a message in chat or mute yourself. And so before we move on, let's just check that everyone's been able to access the R Studio. So please let us know if anyone has any issues. People are quiet. Okay, so I think everyone's good. Okay, so we've already had multiple single cell workshops and talks at this BioC Asia conference. And I think many of us will all be aware and be familiar with this technology and have either generated, used, or read about this type of data at one point. And since its very humble beginnings of genome-wide sequencing of one cell back in 2009, single-cell technologies have advanced a lot, and the number of cells we can now profile at once has grown exponentially. So our ability to profile the transcriptomes of single cell individually has enabled us to dissect the heterogeneity within a population and ask many new biological questions that weren't possible before. And we're now able to appreciate the complexity of biological systems better. And what's more is triggered the development of many single cell specific computational tools that has enabled us to answer these questions. But what's increasingly becoming clear is that single-cell RNA-seq is sometimes not enough. A recent study by Island Klein group asked an important question in their study of hematopoietic stem cell differentiation. And they asked whether the decision of cell fate, as in what the cell becomes later on in their lifetime, can be fully explained by single-cell RNA-seq. And their findings showed that, perhaps unsurprisingly, that this is not the case. They found up to 60% of cell fate decisions can be explained by RNA-seq, but 40% could not. So this really isn't surprising, as we know that cell systems are extremely complicated, and a cell's phenotype is brought about as a result of the interconnected and complex interactions of various cellular properties, such as chromatin accessibility and protein expression, as well as its stochastic events and its interaction with the environment. So I guess in response to this, the next step after single cell analysis has naturally been multimodal analysis to try to gauge what other cellular properties contribute to the overall phenotype of the cell. And multimodal technologies in the strict sense profile 2 or more omics simultaneously at a single cell resolution. Many technologies have been developed for this, as you can see on the screen. And most of these are really at an early stage of development. And some technologies have profiled even 3 to 4 modalities at once, which is quite impressive. And our focus today will be CITE-seq. So the number of CITE-seq data is becoming more and more prevalent. So at the time when we started to think about developing a package for CITE-seq data, there were only very few public data sets out there. But within the past year, there have been many published data sets now for CITE-seq. And here I've just listed very few representative papers. So this also is a testament to how useful this technology is becoming and is also viewed by biologists. So what is CITE-seq? CITE-seq is measures cell surface protein expression. So we're not talking about intracellular protein expression, we're talking about cell surface protein expression and RNA expression of a single cell. It's a technology that is compatible with microfluidic approaches such as 10x, which also means that currently only shallow sequencing can be obtained using CITE-seq data, CITE-seq technology. And specifically, mRNA is profiled in the same way transcriptomes are profiled in a regular microfluidic experiment. But to obtain the cell surface protein expression, an extra step is performed before cells are loaded onto the 10x machine, which is cells are stained with these antibodies against a specific cell surface protein, incubated and then washed prior to sending them through the microfluidic channel. And importantly, in cases where samples are pooled into different samples using hashtag oligonucleotides, we also get matched hashtag expression. And this means that CITE-seq data can potentially give us up to 3 layers of joint information. So what Yingxin and I did, we developed a way to analyze the multimodal data and CITE-seq data in a way that provides us additional information about the phenotype of cells. And we do a host of things in our package, as you can see on the screen, from doublet detection through to many downstream analytical components. And in each case, what we really tried to do was to make maximal use of the multimodal data as much as possible. So the data set that we use in the package and also in our manuscript is one of the first CITE-seq data sets out there, which is from this paper. What they have is PBMC data for CITE-seq. And in the paper, they have other combinations of CITE-seq data as well. And in our paper, we use specifically the control samples. And an integral part of our CiteFuse package is the single cell experiment object. And a key point here is that the single cell RNA-seq data is stored as a primary data, and we store ADT and H2 data as the alternative expression data. And as for any single cell experiment object, the columns denote cells and the rows denote genes. Okay, so let's now move on to the workshop analysis. So processing and integration of CITE-seq data. So the first step is doublet detection. So doublets are unique artifacts of microfluidic approaches where we accidentally co-encapsulate 2 or more cells in a droplet. So this gives us hybrid transcriptomes that we obviously don't want. So we developed a doublet detection approach that is quite specific to CITE-seq and performed a stepwise search for within and cross-sample doublets using both RNA and H2 expression. And we also perform integration of the 2 modalities using similarity network fusion, which essentially integrates cell-cell similarity networks generated from each of the modalities after many iterations until the matrix converges into one. And the resulting fused matrix enables us to do many things such as dimension reduction and clustering. Okay, so are there any questions now before I change screens?
So how do you normalize the ADT data?
Great questions. So we use log ratio transformation of ADT, and that has been the method that the original developers of CITE-seq use. So we use the same normalization approach.
Okay.
Great.
As I can remember, in Surat, they use a different normalization method.
So the last time I checked, they did, from memory, they use centered log transformation. Maybe Yingxin, do you remember what they used?
I think it's different from the RNA-seq normalization.
Yes.
Right?
Yes. Yeah. Okay. So they're different. So we're using the normalization that we also use in CiteFuse is different between ADT and RNA.
Right. Okay. Thank you.
All right. So I hope everyone can see my screen, and I'll just increase the, so thank you for the question, by the way. So many questions are welcome. Just ask questions whenever you would like. Okay, so I hope everyone can see the screen. So if you already haven't done so, we can start by loading the packages that are used in this workshop and the analysis for CiteFuse. So in our CiteFuse package, we have a dummy dataset, which can be extracted using this line of code. And so if you were to generate a CITE-seq data, what you might potentially have is RNA count, ADT count, and also HTO. So once the data is saved as a list, as we can see here, so it contains all the three, the HTO matrix and the ADT matrix here, and also the RNA, where, as I mentioned before, the columns denote cells and the rows denote genes. Here we're using human genes. What we can do is check the dimensions of the data. So with the regular single cell RNA-seq data, we generally have 20,000 plus features denoting genes. And here we have a very simple data set of only 500 cells. And for the ADT, generally it's much lower than the number of features in the RNA. The biggest ADT data set we've seen so far is the recent Bioca paper by Surat version 4, and they had roughly 230 ADTs, which is a massive number of antibodies. So that's the maximum we've seen so far. So the actual dimensions is much smaller than RNA. And the HTO here refers to the hashtag oligonucleotides, and this refers to the number of hashing antibodies that have been used, in which case here there are 4, and this generally refers to the number of samples that have been multiplexed. And as unique for CITE-seq data, the cells are matched. So the expression of the RNA, ADT, and HTO are all matched, meaning that the order of the cells for each of the matrices should be the same. So what we can start doing is to process the 3 matrices into a single cell experiment object. And we do this by using the pre-processing function in CiteFuse. And this generates a single cell experiment object where we contain the RNA count here. And in the alternate expression, we have the ADT and HTO. So the first stage is doublet detection. So we use this log ratio transformation to transform the HTO expression. And we do this and feed the log counts into this single cell experiment object. And the first thing we would do as a quality control after running a CITE-seq data is to see how the cells cluster on the basis of HTO expression. So we can run t-SNE based on the HTO expression. Yep. And scale using PCA. And when we visualize this, using the visualize dim function, which is a very easy function that we developed to visualize the expression data, what we see is that there are 4 distinct clusters. And this is what we would expect, given that there are 4 H2O tags and 4 samples that are multiplexed. Okay. So if we just go back to looking at, I just want to make it clear because sometimes people might not be familiar with the H2O data. It's relatively new. So here, this is what it looks like for the H2O. Usually the number of rows correspond to the number of cell hashing antibodies that are used, and the columns refer to cells. So it becomes really clear when we look across by column that the first cell was a sample that originates from the 4th sample, and the second cell comes from the first sample. And going through the list, we can see which sample each of the cell originates from. So what we would expect is that a single cell should originate from 1 sample only, because they can't be found in both places at once, obviously. So an approach that we took to detect doublets, so when a cell is found to be in both samples, is to use this function called cross-sample doublets. And what we do here is we use mixture modeling to find any cells that seems to express 2 or more of these cell hashing antibodies. So we run this using the cross-sample doublets. So obviously to run this function, you will need to have the H2O expression matrix. And after this, after this function is run, we can go into the cold data of the single cell experiment object to see what the labels are. So based on the mixture modeling, we find these number of cells that originate from each of the hashtags. So 1 here would refer to the H2O28 5P and so forth. And we were able to detect 43 potential doublets that contain 2 or more across 2 or more of these H2O expression. So we can visualize this on the t-SNE we generated before, and then this round, what we can do is to color by the doublet classification. And what becomes really clear is that the single cells sort of found on the periphery of these clusters have been identified as doublets. So yeah, so if you're interested in exactly how the H2 expression looks like for the doublets, what you can do is you can use this plot H2 function to generate a scatter plot of the H2 expression of 2 hashtags at a time for all the single cells. And because there's a sort of, these single cells here seem to express double the amount of H2O hashtags, it becomes really clear that these samples refer to doublets. Okay, and then so we can't stop here because the cross-sample doublets are pretty easy to find because any cells that express this and another cell hashing antibody become really evident that it is a cross-sample doublet. But what would happen, with the same probabilities, we can find single cells that may be co-encapsulated within the same sample. So to detect these types of doublets, we use DB scan clustering based on H2 expression, on RNA expression, to find cells that express sort of double the amount of RNA and also cell hashing counts. So this is a stepwise approach where we first find the cross-sample doublets and then identify the within sample doublets. And these are the number of within sample doublets that we find. And in a similar manner, we can visualize these doublets on the t-SNE. And because they're within sample doublets, it is more difficult to, they're not really found on the periphery, and they tend to be found within the actual H2O classes. And that's what we observe. So finally, what we can do is to filter out the doublets, both the within sample and between sample doublets from our single cell data. And to do this, we just select for the singlets for the within and cross sample, and we filter them. Okay, so with the processing done, what we can now move on to is the fusion of the two matrices. So we perform normalization of the RNA-seq data using the log norm counts function from Skater. And again, as we did for the H2 expression, we perform the log transformation for the ADT expression as well. And what we use in CiteFuse is we apply similarity network fusion to merge the two similarity matrices derived for ADT and RNA using this function called CiteFuse. And because currently we only have 500 cells, the algorithm is pretty quick. But when you have more and more cells, it does take a lot more time. What our function does is it saves the fuse matrix as an SNF_W object. Okay, so with these fuse matrix, we can do a lot of things. So one of the key sort of downstream, one of the key tasks that we would like to do in single cell data is to cluster cells. So what we can do is to use spectral clustering using this function, and you can set the number of K to different values if you would like. And we can access the similarity matrix, the fuse matrix, through the metadata here. And this will be a cell by cell matrix. So we have the same number of columns and rows denoting the total number of cells that we have. And you can plot, oh, sorry, I didn't run this yet. And by plotting the eigenvalues, we can calculate the most optimal number of clusters, which is 5, and run the spectral clustering again. So if you're interested in seeing what the effect of the fusing of the integration of the two matrices are, what you can try is do clustering on the original matrix, the ADT and RNA, and see what how the clustering outcome differs. So for the clustering algorithms, how we designed it is we run the clustering algorithms, and what you would have to do is to manually extract the labels and insert them into the cold data of our single cell experiment using this $sign. And using our reduced dim SNF function, what you can do is to extract the coordinates of the reduced dimensions for t-SNE and UMAP. So at the moment, we allow t-SNE and UMAP reduced dimensions. And again, using the visualize dim function, we can see what the reduced dimension looks like. And each of the plots here are colored by the clustering when it's done on the fuse matrix and the ADT matrix and the RNA similarity matrix. And what we show in our paper and what we can see here is that depending on what type of matrix that you use, the clustering outcome is different. And there are other ways, if you would like to highlight a particular gene expression, you can also color by different expression of either the gene or the ADT to see whether the right marker genes are expressing for your cluster of interest. And we also implement different clustering algorithms, Louvain, from the iGraph package, which can be run using this function. And the same thing can be done. So we don't have to generate the reduced dimensions again, because we did that for before. And this round, what we do is highlight by Louvain clustering. And if you would like an alternative visualization, we also generate this sort of graph as well. Okay. So this is everything up to the pre-processing, integration, and the clustering that's performed in CiteFuse. So unless we have any questions, we'll move on to the next section of the workshop. Any questions? Okay. So I'll give people some time to ask questions. Otherwise, I think Yingxin we can have a switch and we'll move on to the downstream analysis now.
Can you see my screen?
Yes.
Okay. Okay, so next I will introduce some downstream analysis functions in the CITE-seq data or CiteFuse. So one function that we implement in CiteFuse is to check how useful the ADT it is. So this is quite useful when the researchers want to identify which ADT is important to discriminate the cell type identity so that they can keep these proteins in the future experiment. And meanwhile, they can remove those uninformative ADTs in their future experiment. So we use a random forest model to evaluate the importance of each ADT towards the clustering results, and to derive the feature importance as the ADT importance score. So a higher important ADT scores will indicate that this ADT is more discriminative towards the cell types. And another downstream analysis we implement is the ligand receptor interaction. Since the receptors are mostly like proteins, so using the cell surface proteins to quantify the receptor expression of cells would be more could be like more accurate than using the RNA-seq. So in CiteFuse, instead of using RNA-seq as receptor expression, here we use the protein expression to quantify the receptor expression of the cells to calculating the ligand receptor interaction. So similar to what implemented in cell phone DB, we perform a permutation test to select the significant ligand receptor pairs for a pair of clusters. So next I will show how to do it in using the CiteFuse package. So, so also CiteFuse actually includes some of the DE analysis functions and also other downstream analysis function. So for DE analysis, we implement, so currently we only have wheel custom ransom tabs. So we can do the DE analysis for, okay, make this bigger. Is the font size okay now? Okay, yeah. Yeah. So we can perform the DEs on the RNA expression and as well as in for the ADT expression. So here we only need to specify if it's performing to the RNA-seq, we just set the alternative expression as none. And then we also need to specify what group we want to perform the DE. So here we perform the clustering, the low-end clustering results. And yeah, so this sometimes take a minute. And then also we can use the select DE function to select the top DE genes for our future visualization. So here is the DE results for the RNA-seq. And then we can perform using the SAM function to perform the DE analysis for the ADT expression. So we just need to set the alternative name as ADT and then perform the SAM function. So we also have like some visualizations for the DE results. So one plot is the circle pack plot. So we will group the results based on different clustering. So the red indicates the ADT, DE, ADT like differential ADT, and then the blue indicate the differential RNA genes. And we also have the DE comparison plot where we can show that how the p-values are different in ADT and RNA-seq. So here we can compare the DE results of the ADT, for example, like CD4 in protein expression and then CD4 in RNA expression, and compare how they are like have the different DE results. So for example, like here for group 3, in RNA, this is like differential express, it's like a marker for group 3 in RNA, as well as markers for in the ADT. So next, I will introduce the ADT importance evaluation. So this is implemented in the importance ADT function. So here the group indicates that what kind of the grouping we want to see how ADT can discriminate this outcomes. So here we use the low-end clustering results and see what ADT contributes the most in discriminate these clustering results. And we also implement two visualization method to visualize the importance of the ADT. So here we can see that CD4, CD8, CD27, CD5 are the top informative ADT in this data set. So they are mostly powerful in discriminate our clustering results, while some of the ADT, they are like not powerful in discriminate our results. So, and the heat map here indicates the correlation of the ADT, and then here highlight the importance score. So we can see that this part of the proteins have the highest importance in this data set. So next, we will try to remove some of the uninformative ADT and then perform the clustering results again. So this is like pretending in the future experiment that some researchers may want to exclude some of the ADT to save the budget and include some new ADT. So we can see that what happens if we exclude this ADT. And then we will perform the CiteFuse analysis again. So we will perform the integration and also the clustering again with the ADT, with like filtering out the ADT that are uninformative. So we can see that comparing to the original clustering results that using the full set of ADT, using a subset of ADT still have a very high concordance in the clustering results. So the adjusted rank index is evaluating how consistent these two result it is, and we can see that 0.93. So it's like the score is like closer to 1, means that it is like high concordant. So we can see that this is showing a way to how to evaluate the importance of ADT. So next, I will show that how we can do the ligand receptor analysis using CiteFuse. So here we need to input a ligand receptor pair of subset. So here we just save this in the CiteFuse data. So this is a subset data from a cell phone DB database. And then because ADT and RNA-seq has very different distribution, so here we will use scaling method that we think that will make the RNA-seq and ADT have a similar distribution is to scale the data into 0 and 1 ranges, and then we will perform the ligand receptor test using the similar formulas that are implemented in cell phone DB. So this could take a little bit longer time because it is performing a permutation test. So any questions while we are waiting for this permutation test? Okay, just while we are waiting for the functions running, oh, it's actually finished now. So I'm just asking people whether you have like analysis the CITE-seq data before. Maybe click like yes and like some if you have like analysis CITE-seq data before.
So I just started a poll. Question, if you type question 1 means you did, question 2 means you didn't. Maybe you can do that way. Anybody did it? Just type 1, yeah.
Thank you. That's great.
Okay, give me some time. Can't do it myself. Thanks. Seems like a majority of them, you guys have done something. Some of them didn't. Maybe give some background, it's good.
Oh, that's great, actually, yes.
Thank you. So I'm going to end now. I think the voting rate is 50% plus we are in the hosts, you know, administrators.
I vote for that. I vote for it.
Okay, I see. So we're going to end now, okay? If anybody want to vote, that's last call, final. Okay, thank you for polling.
Okay, yeah, thank you.
You're welcome.
Yeah, and then next we can visualize our ligand receptor results. So using this with ligand receptor function, we can visualize, for example, the p-values, whether it's like significant in the rows here indicates the cluster. So is, for example, this cluster 5 as ligand cluster and 6 as receptor cluster. And then the column here indicates the ligand receptor pairs. So for example, this ligand receptor actually is significant in quite a lot of the cell types pairs. So we also have like other kind of the visualization. So if you click on each of it, you can see like it has like different visualization. So we also have like other analysis that you can do with CiteFuse. So for example, we have we have like between sample analysis. So for example, if you have two conditions in your data, you can use this function to analyze your data. And then we also have a gene ADT network. So it's a correlation network that it shows the gene expression and the protein expressions correlation, and then it builds a network. So any questions about the package?
So we have one question. From Julie Zhu, do we have to cluster cells first before performing permutation test?
Yeah.
Yes.
So this is we use the same implement of the cell phone DB. So they compute the ligand receptor based on the cluster results, and so they do we do kind of the average of the based on the cluster for the ligand and receptor expression.
We also have another question. Does the tool support differential abundance analysis? This is a great question. So at the moment, we haven't incorporated information about differential abundance, but I think with more CITE-seq data with multiple samples, I think this definitely would be an interesting or a useful tool to add. Yeah, but currently we don't support that. Yeah, we'll definitely look into that. Thank you. Okay, so I think we have 10 minutes or 5 minutes left, I think.
Maybe 5 minutes.
Yeah. So does anyone else, so we would be happy to take further questions regarding the code analysis. We've been having very good questions so far. Be great if you could also share insight on the experiences that you've had with multimodal analysis as well.
Can I just ask you a question instead of typing?
Of course.
Yeah. So yeah, basically I have a question. So we are doing we're using 10x feature barcode. Barcoding. Yeah, right. So we use this BioLegend feature barcoding. Yeah, right. You know that. So yes. It actually has like in our panel, it has 4 I think isotypes.
Yeah.
Isotypes. Do you consider? I mean, can we use those isotypes for the normalization of the ADT data?
Yeah, so that's definitely something that can happen. So in our package, we didn't actually utilize the isotype information, but with the different isotype antibodies being used, it can also be implemented to account for the specificity of the antibody binding. There are other methods that we're going to touch upon this later in the discussion. There are other methods that are being developed to normalize this ADT expression data. But I do agree, using isotype antibodies, it could be a good option.
Yeah, I asked the 10x people, they said they mainly use those isotypes for background correction.
Yes, yeah.
But I think is another way you probably can use those as a like a normalization, right?
Yeah,
as a
yeah.
Factor. Okay, thank you.
Should we move on to wrap up and conclusion now? Thanks for the great question. So we have some related discussions relating to what Lei said. So we can move on, I think, yeah.
So I will have a quick wrap-up. So CiteFuse can also be performed on the other single-cell multi-omics data, such as the recent germ profiling, Cystic and protein expression data, CITE-seq. So this data is, instead of germ profiling RNA, it's profiling the Cystic with the surface protein. So we performed the Cystic integration and clustering on the aseptic data. And we found that compared to using a Cystic or ADT only, with using CiteFuse procedure, we can obtain a better separated clustering result and like a further annotations that it is not achievable with only using one modality. So we are also aware of some other outstanding questions in CITE-seq data analysis that we are currently working on and trying to implement it in the CiteFuse package. So for example is how do we evaluate the information that gained from RNA-seq and ATAC-seq? And also is how should we normalize the protein expression data from CITE-seq data? So there is a package called DSP that performs the normalization of protein expression based on the empty job list. And we are trying to incorporate this procedure in our CiteFuse package. And also how to deal with the batch effects across the modalities, and do cross modalities suffer from the same degree of the batch effect. So from what we observe is that ADT tend to have a higher batch effects than the RNAs, probably mainly because they have a stronger signals. And we also working on how to deal with the batch effects at the same time and perform the integration afterwards. So for more information about CITE-seq, you can find our paper publication in bioinformatics, and also our package is available in Bioconductor. And if you want to look into more details about how to use our package, you can visit our website that in CineBio's GitHub. So lastly, I would like to thank Cine Precision Bioinformatics Group, and I would also like to thank our supervisors, Hani Jieun, and also our co-authors, Tom. Thank you. And any further questions about our package, you are welcome to stay and ask.
Sorry, I have another question.
Yeah, that's great.
Yeah, so for the ADT data, if you do the batch effect correction, currently, what are those methods you use? I mean, can you use just like a RNA-seq data integration methods?
So, yeah, I'll let Yingxin talk.
Yeah, so I tried using the RNA-seq method to integrate, and I think some of the methods do work quite well.
Such as?
Like Suret, MN, like those. So I think you could like try a bit and see which one works the best. That's my recommendation.
Thank you, yeah, for the insight. So one last question, I promise. So have you compared those, like you have those ADT panel, the number of those antibodies, how they affect your data analysis? Because I mean, theoretically, you can have like hundreds of those antibodies in your like ADT, right?
So we did find that to achieve the same clustering annotations, you don't need the full antibody set. That's what we found. But I think when you do want to dissect the heterogeneity within subpopulations of clusters, having those additional antibodies will help. But in terms of clustering, I don't think having more would
Better.
add that much information because there's a lot of redundant information perhaps. But perhaps when you zoom into one subcluster, perhaps having that extra information would help in terms of analyzing, exploring the data.
Thank you. This is a great tool. I'm definitely going to try it out.
Thank you. Thank you so much. Thank you for your questions as well. Okay, so thank you everyone for attending, and we've had really good questions, and I hope you enjoy using our package. Thank you.